Well, my dear students, let's start our the new chapter now. There is a 12th chapter. Well, there is nothing but biotechnology and its applications. 12th chapter, biotechnology and its applications. In the 11th chapter, you are studying about biotechnology principles and processes. In that, you might have understand the definition of biotechnology, the historical background of biotechnology. If you understand that chapter thoroughly, completely, it will make you easier to understand this chapter, because in that chapter, that is 11th chapter, you study about the principles and processes, the techniques involved in the biotechnology, right? The methodologies, the processes, the experiments that are involved in biotechnology you study in that chapter. Whereas in this chapter we are supposed to discuss about the application part. What is biotechnology? You are studying there. Why biotechnology? You are studying in the 11th chapter. And how is biotechnological processes? All these three questions, for them the answers you are studying in 11th chapter. That is what is biotechnology, why is biotechnology, and how is biotechnology, you are understanding there. In this chapter, the significance of biotechnology, the importance of biotechnology, why do we need biotechnology, and what are the applications of this biotechnology, how do you apply these in order to enhance food production, in order to produce more proteins, enzymes, hormones, chemicals, drugs, pharmaceuticals, right? Medicines, right? In what way it will be useful to mankind. That's why this whole unit is biology and human welfare. Fine. This is the last chapter of that unit. Fine. Fine. So 12th chapter, biotechnology and its application. So before we go into the actual aspect of this chapter, let's try to understand the background of this. Right? In 1919, Carl Lerke gave the definition of biotechnology. You have studied that. Right? Then, why do we need biotechnology? Is a need of the hour to meet the demand of the population as far as food is concerned, as far as medicine is concerned, as far as the other products, biological products are concerned. Biomolecules are concerned, like proteins we require, vitamins we require, hormones are very much needed. Fine, clear. Medicine is required, drugs. Fine. In this application chapter, in your PUC, they have kept two major fields. One is agricultural field, other is a medical field. So in this chapter, what you are going to study involves, fine. One is application of biotechnology in agriculture. So here we talk about genetically modified plants, we talk about transgenic plants, we talk about their therapeutic uses, we talk about their medicinal properties, we talk about their medicinal usefulness, how to overcome the problems that exist in the hybrid plants, the plants that are obtained from somatic hybridization, the plants that are obtained from plant tissue culture, fine. All these problems, to overcome those problems, fine, and to meet the demand of the population, because population is increasing in a very higher degree, fine, very fast, very rapid increase, and to meet the demand. In the previous chapters we have studied about the green revolution. Green revolution has helped a lot to overcome this problem and to meet the demand, but it has not completely bridged the gap. The gap is huge here. Production is at this level, requirement is at this level. So to bridge this gap, to fill this gap of demand from production, green revolution has partially filled this gap, has done, but not completely. So here is the answer: through transgenic plants, through transgenic crops, genetically modified crops, genetically modified fruits. Now what do you mean by genetically modified? Where their genome is being modified. Fine. In the other procedures, in the hybridization, their characters were being utilized. Here you are work, there you have worked at the phenotypical level. There you have worked at the physical level. Here you are manipulating. You are manipulating the genes. You are in this chapter. You will be understanding how to work at genetic level, how to modify organisms, bacteria, genetically, how to change their genes. What do you mean by changing or modifying? Addition of, insertion of beneficial gene, removal of harmful gene, disease causing gene, fine. Any other defective gene, removal of that, addition of a new gene. What is that new gene? Should be a beneficial gene, should be a more productive gene, or that gene should be disease resistant. This gene, if you add into plants or animals, these animals or plants become resistant to that particular disease. And if you are adding this gene, the production has increased, yield has increased. Such a gene if you are adding by transgenic. What is transgenic? The word transgenic, transfer of genes, transfer of gene. You can add the genes of animals into plants, or plants gene into animals, or animals gene into human beings, or plants gene into human beings. Why? Because in your molecular biology chapter you have studied one concept, that is genetic code is, yes, what? Universal. Hence, since genetic code is universal, you can change the genes. You can add any gene from any source, from any organism into any other organism. Why? Because genetic code is universal. I hope it's clear. Molecular biology you have to understand biotechnology, right? Because we deal with genes here, and about genes you have studied in genetics, and about that you have studied in molecular biology also. So those two chapters acts as the base to understand this chapter, and then also the 11th chapter also acts as the base. All these chapters are the background, and this is a application part. So what is the word here? Transgenic means, you might have heard by now many times that transgenic crops, transgenic plants, transgenic animals. So what do you mean by that? What do you mean by that? Where the transfer of genes have been done. Why should we transfer? In order to increase their production, in order to make them disease resistant. Plants are susceptible to many diseases. You have studied these topics in 10th chapter also. So here is a connecting link between all these microbiology and molecular biology. Molecular biology, microbiology combines to give you biotechnology. Biotechnology, it's a complex science, it's a compound science, it's a new branch, it's a composite branch, and it's an applied part. So here you study transgenic plants, agriculture. We talk about transgenic animals also. Partly a small topic is there. After that, applications of transgenic animals. How do we use these animals in a better way for a human welfare, for the betterment of the human beings? How best we can use these animals? How best we can modify these animals? In what way we can change their genome so that we make these animals more productive, more advantageous, more beneficial. In this aspect, this chapter takes you to a new world of application, to a new world of genetic science, where you will be playing with the genes, trying to manipulate the genes, change the genes, alter the genes. So you study first aspect in this chapter, that is application of biotechnology in agriculture. Then in that we talk about the conventional methods first, then we talk about I am giving just introduction to this chapter. After that we will talk about the genetically modified crops or transgenic plants. Then after that in this chapter we talk about transgenic animals. How we have changed the genome of these animals in order to make them more beneficial, more useful to mankind, more useful towards the environment, more useful towards the maintaining of ecological balance in the environment, in the ecosystem, to be more beneficial about transgenic animals. Then transgenic animals, some of the products of these transgenic animals like their proteins, their proteins and their antibodies are used in medicines. Fine, we'll talk about that. Then the second major application of biotechnology is in application of biotechnology in medicine, in medical field. So we have come across here production of human insulin, or we call it as humulin. We have to talk about that. How we produce human insulin from human cells only, from human genes only. These genes are taken, inserted into any other organism. Fine, in detail we will discuss later when we talk about that. So there the insulin is produced and that insulin is given back. Now the question may come, why human being genes? Why human beings? Fine, this insulin is not produced. It is produced normally, but in abnormal cases or in abnormal people, or where this autoimmune disorder is there, where our own beta cells are damaged by our own immune cells, by our own antibodies, fine, or genetic disorders, fine, or inherited characters that leads to the less production of hyposecretion of insulin. Such people were taking earlier the other insulin, but now human insulin is being produced externally. Same insulin is being purified and given back to human being. So we talk about production of human insulin. Very important topic. But there is nothing but production of human insulin. Followed by one more very important topic in case of medicine is gene therapy. You might have heard this in cancer also. I have mentioned one time. In cancer we have mentioned about chemotherapy, immunotherapy, radiotherapy, then we have talked about gene therapy, then surgery we have talked about. What is gene therapy? Fine, very simple to understand. Fine, when we take up this topic, I will tell you. Fine, William Anderson French in 1990 demonstrated gene therapy, where you are manipulating the genes, you are removing the genes. Fine, in the fetal stages, so that these genetic disorders, these genetic diseases, these problems should not phenotypically occur, should not be present in a human being. So we remove such genes, disease-causing genes, through gene therapy. Disease-causing genes are manipulated, suppressed. You have studied this suppression, dominance, right? All this terminology you have studied in Genetics chapter. So here we study about gene therapy also. After that gene therapy, we have small topics like biopatent, bioethics, right? Biopatent we have, bioethics we have, right? Then biosafety measures we have to talk about, ethical issues we have to talk about. So thereby this chapter will be completed. Now, before, this is just what we study overall in this chapter. So before we go into this chapter, fine, what this chapter will tell you about, will tell you how to go about in this chapter, means to say that, fine, we require three major points we have to understand before we go into the chapter. That is nothing but, fine, one is known as biocatalyst. The other is optimum conditions. Optimum conditions. The third is downstreaming process. Downstreaming process or processes. These three terminology we will try to understand today. Fine. So this is the base to understand this topic. Right? Biocatalyst, you have studied catalyst. In chemistry also you have studied. In biomolecules also we have mentioned about this. What is a catalyst? Any compound, chemical expression I am telling, any compound that regulates the rate of reaction without undergoing change in itself. Such a compound, such a molecule, such a substance we can call as a catalyst here. So why we are calling them as biocatalysts? Because these catalysts are obtained through microorganisms. Which microorganisms? The organisms that are being modified, genetically modified. Such organisms are known as GMOs. What do you mean by that? Genetically modified organisms. Genetically modified organisms. If we talk about bacteria, we say genetically modified bacteria. Or on the whole, in general, we talk about genetically modified organisms. If same thing is done, if same manipulation is done, gene manipulation is done in plants, we call them as transgenic plants. If it is done in case of animals, we call here as transgenic animals. Whereas in case of microorganisms, bacteria, we call them as genetically modified organisms, GMOs. What is known as GMOs? What do you mean by that? Genetically modified organisms, where their genes have been modified. In what way? In a more beneficial way. I want to relate you here one molecular biology concept. You have studied about point mutation. Point mutation, because of the sequence of one unit, fine, there will be a change. So such a change is known as point mutation. Fine. So one change in a genome clear, will result in a very different way. Phenotypically, it will be expressed in a very different way. Fine, clear. So one gene is manipulated, one gene is removed, the whole organism will become more beneficial. For example, in pathogens, in some students it may come to your mind, pathogens. If pathogens, their pathogenicity is removed, no more these organisms are pathogens, no more they can cause the diseases. So such modifications, such beautiful experiments we can do here. What do you mean by that? Did you understand? Pathogens, microorganisms, bacteria that cause diseases. Disease causing factor is removed, that gene is removed, that factor is removed from these microorganisms. Thus, these microorganisms can no more be pathogens. They lose the ability to cause diseases. So such beautiful concepts can be done. The problems can be removed from the root level, from the grassroots level. Why? Because we are working at genetic level, we are working at the genes, we are trying to modify the genes here. So biocatalysts, they are required for the production of biomolecules, for the production of pharmaceuticals, for production of vitamins, hormones. They are obtained from the compounds can be obtained from biologically modified organisms that regulate the experiment here, that regulate the rate of reaction. The process of biotechnology, you have studied in the previous chapter that all these experiments has to be done in aseptic conditions. So here catalysts are used to maintain, to produce the products. Then after that, optimum conditions. I hope you are aware of this word. Many a times I used to use optimum, very much required conditions, suitable conditions to carry out the reactions, to carry out the experiments, to carry out the procedure. Optimum, one is minimum, other is maximum. The required one is known as optimum. Optimum conditions are very much required for in our body also to perform the biochemical reactions. In the Asian chapter also I mentioned about these optimum conditions. Right? So here throughout the experiment, throughout the procedure, throughout the process, we have to maintain optimum conditions of pH, optimum conditions of temperature, acidity is maintained, alkalinity is maintained, pH is maintained. What is the required pH? Same pH is maintained throughout the experiment. So optimum condition. That temperature, suitable temperature for that particular experiment. To carry out this procedure, this experiment, this process, this research, that method to carry out this, what is the temperature required? Because microorganisms will be involved, tissues will be involved, cells will be involved, genes will be involved, many other factors will be involving. That depends on the external factors like temperature. So here, constant temperature, optimum temperature has to be maintained, and optimum condition, other conditions. It can be chemical conditions or temperature conditions or pressure conditions, fine. Then amount can be there, fine. So optimum conditions in order to carry out all the experiments to be maintained. After that, downstreaming process. This will be asked for 1 mark. Define or what is downstreaming? The process of obtaining the products, obtaining the biological products that are being done through genetic engineering, that are being done through biotechnology, and purification of dust. Here, what is that? One is known as obtaining. After obtaining the product, you have to purify. Why? Because in most of the cases, whatever the product you have produced here, that is you are studying in first chapter, 11th chapter, biotechnology. So whatever the product you are producing, for example, insulin you are producing, fine, you have to obtain that, you have to produce that. After producing, it will not be there in a purified form. It will not be available in the readily usable form. So that has to be modified. That has to be purified. Fine. So such a process of obtaining the products from genetic engineering and purification process is what is known as downstreaming. Downstreaming. The definition you studied is in the 11th chapter also. There also you come across. It is one of the major concept of major part of the biotechnological processes. In the 11th chapter you study downstreaming. So the process of obtaining the products, after obtaining them, fine, then purification process. What is the purification process? They are simplified. Whatever the problems, whatever the negative aspects, whatever the side effects in them, whatever the harmful chemicals that are there, that are yet present, still present in that product will be removed, will be taken off, will be purified. After purification, after it is been converted to usable form, given a different structure, shape, so that it can be marketed, any hormone can be marketed, protein or drug. Drug here refers to antibiotics, fine. So any antibiotics can be referred, fine. Any antibiotics can be prepared and used into market, can be applied into animals or the human beings. So such a complete process, whether this process is a general process. These are all general processes that will involve in all the processes, fine, and experiments of biotechnology, irrespective of whether you are applying these products, these procedures in agriculture or in medicine or into any other microbiological field or into any other field, wherever you apply, these three factors are very much required to understand their application part. So after these three methods, the first major topic is application of biotechnology in agriculture. So here, why do we go for? I'll just check the time. So almost 20 minutes over, fine. So we have talked about agro-based agriculture there, conventional method of agriculture. We'll talk about that. The next heading is take down application of biotechnology in agriculture. So this is very simple concept. Only have put few aspects, basic aspects of this chapter. From the next class onwards, we'll directly get into the application part. Fine. Keep studying. God bless you. Good luck. Take care.